Welcome, learners. My name is Mr. Pochilola, your teacher on this platform. Welcome to the platform. This is Black Slides Online, where you learn the sciences, namely physics, chemistry, biology, mathematics, and agriculture. We meet now for a Form 3 lesson. This is agriculture. We are going to dwell on a topic called seed and planting materials. Welcome once again. If you are new students and you are learning on our main platform, learning platform, make sure you complete the tasks that are posted there from time to time. Welcome, learners. So this is Black Slides Online Secondary School, your school. If you are learning on our YouTube channel, consider subscribing to the channel so that every time we upload new material there, you are notified. So subscribe and hit the notification bell so that every time we upload something new, you are notified on time. In case all you want is to join us in our main class platform, just simply send us a text message to the number shared here. Welcome, learners. So without wasting your precious time, let's get down to the business of this time. All right, so let's look at some important terms that are applicable in this topic, in this lesson. We shall define the term seed in agriculture as simply a mature fertilized ovule. A seed is a mature fertilized ovule. It is produced sexually by the fusion of male and female gametes. So we are saying a seed is a mature fertilized ovule which is produced sexually, meaning that there is union of male and female gametes in the process. So seeds like bean seeds, groundnut seeds, maize seeds, all those are produced by a method called sexual reproduction, also called sexual propagation. It involves the fusion of male and female gametes. Remember that gametes are cells, sex cells, but with respect to plants. All right. So let's examine the structure of a legume seed. Seeds such as beans, groundnuts, soybeans, and others belong to a broad family called legumes. Structure of a legume seed. So a legume seed has the following parts whose function you are supposed to know. A bean seed has a part called funicle. A funicle is a short stalk which attaches the bean seed into the inside of the pod, also used as a food passage for the developing seed. So a funicle is this part here. It attaches the seed to the pod and transports food. So make sure where it is located and what it does. So a funicle attaches the seed to the pod. The pod is when you're harvesting beans, you actually pluck that long structure and peel it out, and inside it you find bean seeds, which are attached to the pod. So the attachment is called a funicle. A bean seed also has what is called a hilum. The use of the hilum, basically hilum is a black scar where a funicle was attached when the seed is in its pod. When you get a bean seed from it, the part which attaches to the funicle is called the hilum. So the hilum is this bit here. It's the part of the seed, the bean seed, which is attached to the funicle. And then we have a tester, which is also called the seed coat, which protects the embryo against injury and the entry of pathogens. So when you get a seed, a bean seed, you have that red coating outside it, outside the seed. That's called a tester. So the seed coat or a tester protects the embryo against injury and also against entry of pathogens. Pathogens are disease-causing agents. Of course, this part I have not shown, but suffice to say that the tester is that part which attaches to the seed, between the seed and the funicle. Welcome, learners. And then we have a part called micropyle. A micropyle is a tiny hole which allows air to pass through for the respiration of the embryo. We are looking at this developing seed in this case. So the use of the micropyle is just to allow air and water to enter the seed. This part here is close to the hilum. Welcome, learners. So the micropyle is a tiny hole which allows air to pass through for the respiration of the growing seed, the embryo. And then we have the cotyledons. The cotyledons are basically a pair of seed leaves which contain food for the growing embryo. So this is cotyledon, this one. There are two of them. So when you split open a seed, you see these two. The use of the cotyledon is to store food for the embryo, for the growing plant. So take note that seeds with two cotyledons are known as dicotyledonous seeds, or simply dicots. For example, beans and the groundnuts. When you split open a seed, a bean seed, you see these cotyledons. Di means two. So since there are two cotyledons, we call it dicotyledonous seed. And then we have a radicle. The radicle is part, we have already mentioned that, maybe. But just in case we have the radicle, The radicle is part of the embryo which forms the primary root. Yeah, so this one here. So the radicle develops into primary roots. And then we have the plumule. Plumule is the other end of the embryo which grows into a young shoot. So the plumule grows above the ground as a shoot, develops into a shoot, and it is the one that forms the first leaves of the seed. And then we have the hypocotyl. The hypocotyl is the one that grows into a stem. It's the part of the embryonic structure which develops into a stem. So we have looked at the parts, the structure of legume seed. For example, the groundnut seed or the bean seed. So whether it's bean seed, soya bean, or any other dicotyledonous plant seed, these are the parts. You must know them and be able to respond to questions that asks you to do with the structure of a leguminous seed. Examples of leguminous seeds are groundnut seeds, soya beans, and many others you can think of. Let's look at another kind of seed which is called a cereal seed. Cereal seed, an example is rice, millet, sorghum, maize. The very best example is maize seed. It's a cereal seed which has a fused pericarp and tester. So the pericarp and tester in cereals are fused together. The use of the tester and pericarp is to protect the delicate internal parts of the grain. Welcome, learners. And then there is a point of attachment to the cob, which attaches the grain to the cob. In this case, in terms of maize. And then it has an endosperm, which supplies food for germination and the development of the embryo. So all seeds containing this are called endospermic seeds. So a good example is this one here. So we have a pericarp and tester. The outer part of a maize seed is fused so it contains a fused seed, coat that protects the grain. So it protects the internal parts of the grain. The fused pericarp and tester protects the delicate internal parts of the grain, and then there's a point of attachment of the cob. Endosperm supplies food for germination and the development of the embryo. All the seeds containing this are called endospermic seeds. The radicle develops into a primary root, while the hypocotyl develops into a stem. The plumule develops into the first leaves of the maize seed. Mind you, we are discussing an example of a cereal seed here, which is maize. Then we have rhizosphere, which forms a protective sheath against the roots, enclosing the roots. And then we have a cryoptyre, which protects the shoots as it pushes through the soil. And then we have the sclerum. The sclerum forms the outer rim of the embryo. All right. So this part forms the outer rim of the embryo. Since there is only one cotyledon, seeds in this group are called monocotyledonous seeds, or simply monocots. Examples are maize, rice, sorghum, and others. Welcome, learners. So a maize seed will look like this. So we made reference to the pericarpon tester, which is this outer part, which is basically a fused seed coat that protects the grains, especially the inner parts. And then we have the endosperm, where food is stored, and this food is supplied to the seed during germination and the further development. And then we have a point of attachment, which is this bit here, right? This attaches the grain to the cob. So when you're sharing maize, it is this part which is attached, part of the seed which is attached to the maize cob. And then we have a scrotum, which is a single cotyledon that absorbs food from the endosperm. So this is used for absorbing food from the endosperm. And then we have a premium. Premium develops into a first leaves of the maize seed during germination. And then we have a cryopatile, which protects the premium as it emerges. And then we have the hypocotyl, which develops into the stem. The radicle is that part of the seed that develops into primary shoots. And lastly, we have what is called the cryolysal. The cryolysal protects the radicle. Welcome, learners. Let's discuss a very useful concept in agriculture, which is called the plant propagation. Plant propagation is the same as plant multiplication. Propagation means multiplication. So as we are going to see in this lesson, there are two types of propagation, or there are two major methods of multiplying plants. The first one, which is called sexual propagation. This is the multiplication of plants by using seeds. So in this case, it means the fusion of male and female gametes of the plants to form seeds. All right, so plants such as maize, groundnuts, beans, and others use sexual propagation in order to multiply. Every plant propagation technique has advantages and disadvantages. Let's explore the advantages of sexual reproduction. It's the type of multiplication of plants by using seeds. So one of the advantages is that seeds are relatively cheap and they're easy to sow, like to plant, handle, and prepare for planting. Seeds are easy to sow, handle, and prepare for planting. Seed is easy to store without loss of quality or quantity, as long as you treat them against pests. Seeds can remain viable or alive for a long time, as long as they are safely stored. For example, if you have maize seeds you are planning to plant next year, you can keep them safely and still be able to germinate them during the planting season. Seeds can easily be sown mechanically using drills. So you can make drills on the soil and bury the seeds there. They are going to germinate into plants. Seeds minimize the risk of disease transmission from parents to offsprings. And the seeds offer the only way of propagating crops that cannot be propagated vegetatively. And lastly, on the advantages of sexual propagation, seeds produce new plants that differ from their parents, leading to crop improvement. In case you are not sure about what we are discussing, feel free to ask questions. I'll be glad to share with you where you might miss some points. Welcome, learners. However, propagating or multiplying plants by method of seeds has disadvantages as well. In some cases, it can produce various variations of offspring. Can produce serious variation and of types among the offsprings. So you find that some offsprings differ from each other within the same population. That's a disadvantage. In other words, they lack uniformity of offsprings. So offsprings may not show the exact good qualities of the parent plant. The offsprings may be less disease resistant or pest resistant than their parents. In most cases, propagation of plants by seeds takes longer to grow and mature compared to vegetative propagation, a technique which we are going to look at. Some seeds have low germination rates, are hard to grow. Some seeds may not germinate at all. You find that out of 100 seeds, only 80 germinate and the other 20 do not germinate. That's a problem also. This requires special care for young seedlings in order to survive. So this method is not suitable for plants that produce no seeds or have non-viable seeds. So plants that do not produce seeds cannot be propagated this way as seeds. Welcome, learners. This second type of propagation is called asexual. Asexual, when a is placed before a word, it means without. So asexual means without sex, without exchange of, or without a fusion of male and female gametes. Okay, so asexual reproduction refers to the production of new plants vegetatively. In other words, using plant parts such as cassava, for example. You plant stems, cassava stems. When you are planting cassava, you don't use seeds. Sugarcane is another one. Plants such as ginger are grown this way. Okay, so this is the production of new plants vegetatively. Vegetatively means using the stems, the parts of the plants themselves without using seeds. And this technique is sometimes known as vegetative propagation. So vegetative propagation uses the following materials. You can use stems, so cassava cuttings, which are also called seeds of sugarcane. Cassava are in this type, cassava and others. You can also use runners. This is a type, it's a stem which grows along the ground, like sweet potatoes and star grass are grown this way using runners. Stem tubers, this is where the soil portion of the plant is buried into the ground. For example, Irish potato, you plant stem tubers. Suckers, a sucker is a shoot arising from an axillary bud at the base of a parent plant. For example, bananas and pineapples are grown using suckers. Bulbs, these are made up of closely packed stems, such as, for example, onions and garlic. Cones, this is the thickened base of an underground stem. Rhizomes, they are the thick horizontal underground stems. For example, bamboos. Another technique that has to do with vegetative propagation materials is layering. This is where a branch of a tree or shrub is pegged to the ground so that it drops into roots for new shoots while in the soil attached to the plant. Another one is budding. Budding is when vegetative bud from the plant is transferred and joined to the stem of another plant where it develops into a new shoot. Budding. And then we have grafting, which involves joining parts of one plant to another plant. That's asexual reproduction. Like we saw with sexual reproduction, asexual reproduction or propagation of plants also has advantages and disadvantages. Among the advantages, it is that it reduces the juvenile period. So new plants take after the parent's age. Alright, new plants take after the parent's age. It eliminates seed dominance issues common in many seeds. And it also ensures genetic uniformity. Offspring are identical to the parent. This is the only method for crops that produce non-viable seeds. So planting paths are harder. Meaning that harder than seedlings, meaning that they withstand harsh conditions such as drought, for example. They have stores of food, stored food reserves. For that reason, they are easier for new shoots to establish. These are easily available to farmers from previous harvests. Let's say you just harvested cassava, so you can get stems and plant again. The disadvantages of asexual propagation include high risk of spreading diseases from parents to new plants. There's little natural variation. It makes crop improvement very difficult. They are back and heavy, harder to store, carry or plant than seeds. Some techniques need special training or skills, so these are the advantages, these disadvantages of asexual propagation. So please know the advantage and disadvantage of which method. In an exam, the examiners will ask you to state those. So know all of them so that you can give any two when required. All right, thank you very much. Before I go, I would like you to do these questions for me. And kindly share your answers with me. I'll be excited to see your work. So define sexual propagation. Give 2 examples of dicotyledonous seeds and 2 examples of monocotyledonous seeds. State 3 advantages and 2 disadvantages of sexual propagation. Explain the difference between budding and grafting. What is asexual or vegetative propagation? List 4 methods of vegetative propagation and give one crop for each. A farmer wants uniform crops that mature early. Which propagation method is best? Give 3 reasons. So kindly share your answers with me. I should have a look at them. For the time being, bye bye. We meet again in the next lesson.